So, now let us discuss about the transcription unit. Yes, the transcription unit, it is the unit or a segment of DNA, right. It is a segment of DNA. On that there are 3 regions are find out. So, that is it consists of 3 regions on the DNA, right. There are 3 regions which we identified, that is the on the DNA segment, right. So, first one, the transcription unit have 3 regions, that is first one is a promoter and the structural gene, right. So, the structural gene, right. Next one, a terminator, structural gene and a terminator. These are the 3 regions which we observed on the the DNA, that is called a transcription unit. All are involved in the transcription process. So, as we know, it is one of the heterocatalytic process, because here from DNA to DNA prepare or the synthesize the RNA. Okay. Replication was the autocatalytic. So, this is the heterocatalytic process or phenomenon, right. So, what I have taken here, the schematic representation or schematic structure of transcription unit. So, it may be expected for 5 marks in your examination. So, schematic structure or the schematic representation of transcription unit, right. So, here the 2 strands which are opened. So, actually the DNA may be it is a elongated one, DNA is elongated. So, for our understanding, so we are representing the open 2 strands here, okay, a small segment, for example, this much I have taken here, right. So, this is what the structure, right. So, here the 2 strands are there, as we know they are anti-parallel to each other, and they are complementary to each other, right. So, these are the 2 strands of the DNA, right. So, here the the structures involves the first part, that is called promoter. Yes, it contains promoter. Yes, the first region is called promoter, the next region this one, right. So, this is what the complete region is called structural gene, what we call structural gene. This is the first part is means it is some sequence, we will discuss it later. So, try to understand the structure first. So, this is the promoter region, and next this much is structural gene, and next the end part is called terminator, right, terminator, which contains some sequence, DNA sequence. This is also having DNA sequence, and this is a little more DNA sequences, right. So, now there are 2 strands which we called, one is coding strand. So, this is what the coding strand, which has a polarity from 5th direction to 3rd direction, which is there in this segment, that is called coding strand, which does not code for the synthesis of mRNA, hence it is also called non-transcribing strand, right. And another one is called template. To this coding strand, there is a template strand, that is also called transcribing strand, because so this involved in the synthesis of mRNA to for this as a complementary new strand is formed, right. So, this region is called the structural gene, right. So, this is about the the unit, schematic structure of transcription unit. So, here enzymes are involved, as you know, in the replication many enzymes are involved. So, here also some enzymes are required, especially the DNA dependent RNA polymerase is used. DNA dependent RNA polymerase catalyze the this process, that is polymerization in only one direction, that is in one direction, right. So, in one direction, that is from polarity, that is 5th to 3rd prime, 5th prime to 3rd prime in this direction. So, the polymerization would takes place here with the help of DNA dependent RNA polymerase. Yes, DNA dependent RNA polymerase, because the synthesis in transcription, what is synthesized? So, that is mRNA. RNA is synthesized here. So, that is why RNA polymerase is required. RNA polymerase, it is important. RNA polymerase, which depends on the DNA. So, that is why DNA dependent RNA polymerase we can say, right. So, next about the promoter, let us we discuss about the promoter. So, which is located at the fifth end of coding strand, fifth end of coding strand. So, this is what the promoter is, not that side. So, not to the third end, right. So, that is the promoter is located so at or towards this fifth end. So, that may be extended part, that is why we can take it as towards the fifth end or fifth prime of the coding strand, right. So, which is located, and next these actually the promoter and terminator are present on the either side of the structural gene, right. So, one is with the one promoter is with the fifth end and terminator with the third end. That is what we call, this is called the upstream, okay, upstream, and it is towards terminator towards the downstream, you can say, right. So, next it is a short sequence of DNA. It is a short sequence of DNA. How we observe the primer in case of DNA replication. So, here there is a RNA primer it was, but here, so we will not call it as primer, it is a promoter we called. So, here the short sequence of DNA, right, a short sequence of DNA is present here, usually TA like some sequence will be there. So, hence it is also called Tata box, right. So, Tata box which begins the transcription, which begins the or initiates the transcription like a starter, okay. It starts the the transcription process. Transcription is proceeded by the help of this promoter. So, that is what the short sequence of DNA that provides the binding site, for RNA polymerase, for this RNA polymerase to polymerization, which helps in the polymerization. No? So, that polymerization begins with the promoter because it has a binding site of this what you call RNA polymerase. So, the short sequence that may be TATA. So, that is why we also refer it as a Tata box, okay, which is the what you call identification of the presence of promoter, right, promoter which promotes or the what you call which begins this process. Transcription is begins with the promoter, right. So, next one about structural gene. Let us we start the structural gene now. Structural gene. Yes, the structural gene. So, this is what the structural gene region. So, here the DNA strand having polarity, that is third towards fifth, third to fifth. So, that is called template, that is what we call template strand, or simply we can say template or template strand, which is involved in the formation of a new mRNA, right, synthesis of mRNA, hence it is a transcribing strand, hence it is also called a transcribing strand, right, transcribing strand which has a polarity third to fifth prime, right. So, next the other strand, another strand to this one. So, that is a anti-parallel strand is there, no, one more, that is what another strand, right, other strand having polarity from the fifth to third prime, fifth prime to third prime. So, this is what the polarity of another strand. So, that is called coding strand, right. So, this is what the coding strand, what I have mentioned here. So, this is the coding strand, you remember, this is the coding strand has the polarity from fifth end to third end or fifth prime to third prime, right. This is what the direction of the coding strand, and the template strand is third polarity to third prime to fifth prime. This is what the polarity of the template, right. So, usually template is performing the synthesis of new strand, that is the DNA, right. So, DNA is formed here, right. So, here whatever the DNA is formed here, no, from here it will begins with this side. So, this DNA when forms, right. So, it has codes, right. So, this coding sequence is complementary to the to the template. These are complementary to the template. Complementary means, as you know already, so adenine pairs with thymine in case of replication, so and guanine pairs with cytosine. So this is what the complementary, as we discussed in previous video also. So here instead of this one, uracil will be there, uracil, right. So when you observe this one, when you observe, so template is there, no template. So to this mRNA is synthesized, which is exactly similar except in presence of T. So wherever T is here, so there U will be there, right, on this strand. Remaining sequence is similar. So this is what the complementarity which governs the process, this process, and it defines with the coding strand, not the template strand, but it is complementary to the template, but it is similar to the coding except the uracil. So instead of thymine, there is a uracil on the mRNA, because it is a RNA sequence. So as we know the difference, okay. So here instead of thymine, uracil will be present, okay. In the place of thymine, uracil will be present, whatever the same adenine, guanine, so like it may be, right. So on sequence may be any all the different sequence will be present, okay. For a protein synthesis, triplets are, we will read it as triplets, right. So here the only the same coding strand is similar to RNA, this newly synthesized RNA. So it is exactly similar, but only the difference is instead of thymine there is a uracil, right. So in this way the newly synthesized RNA will be formed, right. And so terminator, once it comes to the terminator, so that will stops the process. That is what it is located on 3rd prime, that is 3rd prime, which is referred as downstream, towards the downstream, right, towards the 3rd prime, which is at the 3rd prime of the coding strand, not the template, 3rd prime of the coding strand. So terminator is present or located at or towards the 3rd prime of coding strand, right. So this is what downstream we call downstream. So downstream on the coding strand. And next, the terminator which stops the process of this transcription. Transcription will be stopped at terminator. Next, which can recognize actually the short segment of DNA. Short segment of DNA means this is also a terminator with a short segment of DNA. The DNA sequences are here. So that is recognize the termination factor, okay, recognize the termination factor, which is also referred as rho factor. So which will decide the whether it will be stopped, right. So usually it ends with the terminator. This process transcription ends with the terminator, right. So this is what about the schematic structure of transcription unit. You remember schematic structure or the representation of this diagrammatic representation of the transcription unit. This is important, transcription unit. So as I explained, transcription unit is consist of a promoter and the structural gene and a terminator. These are 3 regions which are found on the segment of a DNA. You should remember this diagram, and let us discuss the details in the protein synthesis and what is gene and how it is conducted, right. So in the next video we will learn more about this one, right. Thank you, students.